Welcome back. This is Mrs. Vance again, and this is the first part of notes for Unit 12. We are starting to learn about microbiology and the immune system, and so we're going to start off with some notes about viruses, and then we'll move on to bacteria, and then we'll talk about the immune system and kind of put it all together at the end. So lots and lots of vocabulary in this unit, as you can see. Make sure that you are working on vocabulary as you have learned. Knowing the vocabulary is really important. There is a quizzes and a Quizlet in the Unit 12 resources folder that you should find in the weekly folder. And so be sure that you're practicing on that as you go along instead of waiting till the end to try to memorize it all at once. It's really important to pay attention to the words. So we'll start off with learning about viruses. Viruses are kind of interesting. We've had a lot of experience with viruses in the last year dealing with the pandemic, and so learning a little bit about viruses and how they work. We'll also learn some things about specifically about the COVID virus as well as other kinds of emerging viruses that we see in the world these days. So viruses are acellular cell parasites. What does that mean? That means they're not made of cells. Viruses, we have a tendency to talk about them being living or dead or killing the viruses and so forth. Viruses are not living, actually, and because they're not made of cells. They are able to reproduce, but only inside a living cell. They can invade and reproduce in cells. They're not classified in a domain or kingdom. There is a classification system for virus. We'll talk about a little bit about that coming up soon. They're very small. They're generally on the range of 17 to 1,000 nanometers in size, a lot smaller than cells. There are a few of the megaviruses that are getting close to being the size of some cells, but we don't see those very much. They live in the ocean mostly, or they are found in the ocean mostly. They're passive. They're just things. You know, they're some people would say that they're living, but they really aren't living. They don't meet the criteria that we set for cells. They're controlled by their environment. They're at the mercy of the environment, and they're only controlled by their genetic material. They don't really do anything except when they invade a cell. Then they take over the protein, the machinery of the cell, basically, to make copies of viruses, and then they kill the cell when they're leaving. So they're not really anything beneficial. They're just there. A virus is essentially, by definition, genes in a box. Okay. They are infectious particles. They've got some nucleic acid in them. They can have different kinds of nucleic acid genomes. Some of them have RNA, some have DNA, some have single-stranded RNA, some have double-stranded RNA, single-stranded DNA, double-stranded DNA, all different kind of combinations of things. They're wrapped in a protein coat that we call a capsid, and in some cases they're in a membrane envelope, which is usually the ones that are going to be infecting cells. There are two types of reproductive cycles we'll talk about. First of all, the lytic cycle. This is when you have an active virus infection. The viral particles are produced using the parts of the host cell. In other words, they take over the DNA or RNA replication machinery. They take over the transcription and translation. They're making proteins, all this kind of stuff. Once they have made a bunch of viruses and the cell is pretty much full of viruses and nothing else, then the host cell will lyse or burst, and that releases viruses to go infect more cells. So when you have an active viral infection, that's what's going on. You have a virus in the lytic cycle, kind of like this. Okay, the diagrams that we're showing here and that we'll continue to talk about are talking about a specific kind of virus called the bacteriophage. Those are the first ones that were studied intensively to learn kind of how viruses work. There are specific viruses that specifically infect bacterial cells, and so we'll talk about those as well. So in this case, in the lytic cycle of this bacteriophage, the virus attaches to the cell. In the case of bacteriophages, the bacterial cell doesn't take the whole virus in, but the virus actually injects its DNA into the cell. The phage DNA kind of takes over and starts causing the cell to make new copies of the phage DNA, as well as the proteins necessary for the capsid. The phages get assembled automatically. They're kind of auto-assembly programmed, and then once the cell is full of viruses, then the cell lyses, and that releases the phages, and then they can go attack other cells. Realize, please, that the scale of this is a lot different than it shows here. It's just difficult to show relative scale. These are much, much smaller than the cells. And so you can get hundreds, if not thousands, of viruses inside the cell, even in something as small as a bacterial cell. In the lysogenic cycle, the viral DNA gets inserted into the host chromosome rather than just being inserted into the cell. And the viral DNA is replicated just every time the cell undergoes cell division, the S phase of the cell cycle replication, then it's going to be copied along with the host chromosome during each cell division. In the inserted viral DNA is called a prophage. In the case of the bacteriophages, it's called a provirus. In the case of animal viruses, most of the viral genes are inactive. And so it just kind of goes along, just kind of sits there and hides and waits around. At some point, environmental signals can basically turn on a switch to the lytic cycle, causing the viral DNA to remove itself from the chromosome and lead to the death of the cell via the lytic cycle, like we see here. So in this case, after the phage DNA injects, it actually becomes inserts into the bacterial chromosome by recombination, and then it gets copied every time you have a new cell, and then it can go into the lytic phase depending on the environmental conditions at the time. We have similar things that happen in animals as well. If you've ever had fever blisters or cold sores, the kind that reappear around your mouth, that's a virus that hides out and then is stimulated to become lysogenic at some point by some kind of environmental signals. It just depends on the individual. I sometimes get fever blisters. I used to get them a lot more often when I was younger. But basically, they usually appear in pretty much the same place around my mouth. I can usually tell they're coming on because it starts to feel itchy in that area. Sometimes it's by stress, sometimes it's when you get sick, things like that. I used to always break out in fever blisters like shortly before school started because I was excited and anxious about school starting when I was a kid. And so, it just depends on the individual. Viruses are classified by their genome, either RNA or DNA. Some RNA viruses are called retroviruses, and we'll talk about what that means. Some of them have, like I said before, single-stranded RNA, single-stranded DNA, double-stranded RNA, double-stranded DNA. There are lots of different things. They're also classified by what they might infect. For instance, a bacteriophage that we talked about is a virus that infects bacteria. And then they're also classified by shape. And so here we see a couple of the classic shapes here. Spherical. This one looks sort of like a coronavirus, not exactly, because these proteins are a little bit different than you see on a coronavirus, but they're spherical. Polyhedral, meaning that they should have a regular geometric shape. Helical means that you've got capsid proteins that are basically attached to the helical nucleic acid. It looks kind of like a flower when you look at it. If you look at it in one dimension, it looks kind of like a rod-shaped structure, and other times it looks like like a flower. And then we have the binyl or complex combination. This is a bacteriophage that has a polyhedral head, a helical tail, and then sometimes they have these tail fibers. These are not legs. It doesn't walk around. They're just some structures that are there for part of the attachment process. It just depends. So in class, we're going to make a model using some regular things that we could have in class. And so we'll have, in class, I'll have some copies of different genomes, some double-stranded DNA, some single-stranded DNA, single-stranded RNA, double-stranded RNA. That would be the nucleic acid that's going to be in there. And so the students will label that. There will be an envelope like you would send a letter in, and then we're going to call that the envelope. There's also a small Ziploc bag that will label as a capsid, and then there'll be some shapes, various kinds of shapes. Some of them are triangles, and some of them are circles, and some of them are arrows, and different kinds of things like that. And so those are going to be the glycoproteins that are often found on the outside of the envelope. And so the way it's going to be put together is they'll place a nucleic acid inside their capsid, and then put the capsid inside the envelope, and then they'll put their glycoproteins on the envelope so they're hanging over the edge. And I'll give you an example in just a minute. So we're going to do that in class. I'm going to show you what it looks like in just a minute. Like this. Okay, so here we have an example that I made. This is my Ziploc bag that's containing my genome. This particular one is double-stranded RNA. Notice that it's RNA because it's got U instead of T, but it's double-stranded because you have both strands there. So I just labeled it dsRNA. Here's my envelope with my glycoproteins attached to the outside. And so then I put the capsid with the genome inside the envelope, and that makes our completed virus. So how does that work? Well, viruses attach to the host cell because the glycoproteins on the envelope actually attach to receptors on the host cell, kind of like puzzle pieces going together. And if you've paid much attention in the last year when we've been talking more and more about the coronavirus, the one thing that's unique about the coronavirus, or the SARS-CoV-2 is the official name of the virus that causes the COVID-19. It has the spike proteins on the outside, or those glycoproteins, and they attach to a specific kind of receptor in humans called the ACE2 receptor, which is very you have a lot of copies of that in cells of your respiratory tract, inside your nose and your upper throat, and then a lot more in your lungs, as well as in some other organs as well, which is part of the reason why the coronavirus disease, the COVID-19, actually affects so many different organs and organ systems, because there are these ACE2 receptors all over the place in your body. So that's kind of how that works. So let's look at an example. Here are some examples of cells with different kinds of receptors on them. And so if we take our virus that we made a while ago, okay, this one right here, which cells could this virus infect? Well, if we put it over to the edge like this, you can see that that point on that pentagon actually would fit right there in that receptor on that particular cell. If we rotated it a little bit, then the square part at the bottom of the pentagon might fit right inside the receptor there. It doesn't fit exactly right, but it might fit well enough to infect that cell. And so part of it depends on what the cell is, and as opposed to what kind of virus can infect that. And I think we probably understand that a little bit better now, having gone through the last year of this pandemic, than we did a year ago when we were talking about this in class. So we're going to stop right here. That will be the end of the lesson for today.